आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
Vigyan Ki Awaaz podcast series aims to popularize new ideas of science and technology, which highlight STEM, science, technology, engineering, mathematics, and medicine. This brings to the surface the efforts and researches of Indian scientists, which is desired to bring a change in community learning and teaching, access to knowledge of resources, and social paradigms. Our goal is to spread the name of indigenous Indian research stories and their masters among the masses of society in a very easy manner. Bose Institute was set up in 1917 by Sir Jagadish Chandra Bose, the founder of modern science in the Indian subcontinent. It is Asia's first modern research center devoted to interdisciplinary research and bears a century-old tradition. The institute was founded as not merely a laboratory but a temple, with its principal aims being the advancement of science and diffusion of knowledge. Swami Vivekananda, Sister Nivedita, whose influence is reflected in the institute's emblem of Vajra, was significant in the setting up of the institute, and Rabindranath Tagore were among those who had consistently inspired and supported Jagadish Chandra Bose in his endeavors. Ever since the establishment of the Department of Science and Technology, Government of India in May 1971, Bose Institute has been an autonomous grant-in-aid institution of DST. Over the last century, the institute has endlessly strived to preserve the legacy of its illustrious founder. In this podcast, we are happy to present Dr. Jayanta Mukhopadhyay, professor in Bose Institute. Before going into details about his research work, let us hear from his own voice about his academic journey.
I'm a professor at the Department of Chemical Science. I did my master's in IIT from IIT Kharagpur and in physics. And then I switched my field. I started working in biology. So I did my PhD from Bose Institute in biophysics and biochemistry. And then I moved to USA at Rutgers University. I did my postdoc over there. I spent several years. And then I moved back to India and joined Bose Institute and started my own lab.
Dr. Mukhopadhyay shares some details about his area of research.
So my laboratory is involved in understanding the fundamental mechanism of transcription and gene regulation. And we are also involved in identifying new inhibitors for transcription. Okay, so let us first talk about gene regulation in bacteria. So bacteria have thousands of genes. And some of the genes are expressed continuously, called housekeeping genes. But most of the genes are expressed transiently, depending on the bacteria's need. For example, a bacteria attacks you. Now your body has a defense mechanism that tries to kill the bacteria. Now bacteria will employ their survival strategy to combat the attack by the host cell. So this could be achieved by controlling expression of certain genes. So this is gene regulation. So gene regulation is switching on or switching off the expression of genes or controlling the level of expression of genes in bacteria. In bacteria, most of the gene regulation occurs at the level of transcription, the process of encoding DNA into RNA. Now, RNA polymerase is the enzyme responsible for transcription. So, there are some controlling element in the gene called promoters, and there are many proteins called transcription factor that binds near the promoter and either facilitates or obstruct the gene expression. So, this is the way gene regulation occurs, and this is a very complex mechanism. Some of the mechanism of gene regulation are known, but there are many still unknown. So our lab is trying to understand the unknown mechanism of gene regulation in bacteria. We proposed few model of gene regulation in Mycobacterium tuberculosis, the bacteria causing TB, and also in Bacillus subtilis, a model organism of gram-positive bacteria like Staphylococcus and Streptococcus. We published few articles in reputed journals like Nucleic Acid Research, Journal of Biological Chemistry, the Journal of Molecular Biology. So the question is why understanding gene regulation in bacteria is important. So let me explain in this way. If I ask you to kill a person, how do you do it? So you have to target the important organ of a person, like heart, lung, or brain. So if you want to kill a bacteria, you need to identify a target. That means you have to block the important function of the bacteria. A detailed understanding of the transcription will lead to find new target for antibacterial therapy. Now, transcription is already validated target for antibacterial therapy. Rifampicin, the drug used for TB, for the treatment of TB, blocks transcription. However, with time, the bacteria becomes resistant to rifampicin. Now, there is a urgent need to identify new inhibitors that act against the drug-resistant bacteria. So recently we have characterized a novel inhibitors so that function against the resistant bacteria. We identify the mechanism of inhibition and we establishes that this molecule can be act against the drug-resistant bacteria.
Dr. Mukhopadhyay explains in details about the research collaborations.
So most of the work I do alone, but for the drug, what I talking about, so one scientist from Spain, so she published this chemical, she synthesized this chemical and I thought maybe this will be a very useful target for TB. So I collaborate with her and we established the mechanism and we are about to publish this in the journal. So this could be, and we also establishes that this could be a drug against the rifampicin resistant bacteria.
Dr. Mukhopadhyay shares his experience about working in his laboratory.
So in our lab, we have developed the assays, some biochemical assays. So as I told you that RNA polymerase is responsible for transcription. Now in most of the lab, we don't have facility to handle the TB bacteria. So what I do, I take the genes from the TB and express in E. coli so that I don't have to handle TB. And I can purify the polymerase, I can do the assay, and then I see whether the drug is active against this one. Then I develop some RNA polymerase which is resistant to rifampicin. That is happens to a patient. Then we check whether this is working as the rifampicin resistant strain. Then we came to conclusion that this drug is acting against the rifampicin resistant strain.
Dr. Jayanta Mukhopadhyay speaks about the involvement of industry in pre- and post-pandemic era in this field of research.
Let me talk a little bit about history of antibiotics. So the first antibiotic was discovered in the 40s by Sir Alexander Fleming, and that earned him a Nobel Prize. And subsequently, there is a quite few Nobel Prize for the discovery of antibiotics. And the craze of identifying new antibiotics continued up to 70s and early 80s. And there is a lot of pharma company, they have developed new antibiotics. Now the scenario has changed. No pharma company are interested anymore in investing money into the antibacterial therapy because there is no significant profit. So the research on bacteria or antimicrobial agent is being carried out in our lab or lab like ours in academia, no industry. And the funds for the bacteriology also dried down mostly in developed countries. So, however, the COVID-19 is an eye-opener. Now the people have realized there is a urgent need of microbiological research, and we discreetly continue to do whether we can identify new target, new drug. So that is the way we do research. So we have developed assays to identify antimicrobial agents, and we have developed assay to identify inhibitors for transcription. So actually any industry can use our technology, they can collaborate with us, and we are ready to share our knowledge. But as I told you that most of the pharma company, they are not interested in developing new antibiotics. If I told them this is antibiotic, they will do the marketing. So, but hopefully in near future, maybe some company will be interested, and we are ready to be involved.
We are thankful to Dr. Jayanta Mukhopadhyay for providing us with his valuable time.